The visual organs of insects. Photoreceptive sense cells are innervated by bipolar neurons like that of other receptors, but differ by containing fine surface striations rather than a definite dendrite tip, and their overlying cuticle is transparent and form a cornea for transmission and conduction of light rays. These photoreceptive structures group together and form definite visual organs. The most primitive photoreceptive organs of insects are represented by the paired small papillae on apex of the head of the mascoid maggots. These organs resemble the typical chemoreceptors in a structure, but due to modification of overlaying cuticle into transparent biconcave cone of lens, they function as the photoreceptors. The true eyes are found in almost all insects except few parasitic forms and are of two types, the simple and compound. The simple eyes are dorsal ocellae and the lateral stemata. The latter are often found in holometabolous larvae. The compound eyes are the principal visual organs of the adult insects except Sypungulata, Siphonoptera, female coccyps, and some cave-dwelling insects. So in detail, we are going to learn the dorsal ocelli. They are found in the nymphs of hemimetabolous insects and in almost all adult insects. The insect bears three ocelli, one median and two lateral, forming a sort of inverted triangle anteriodorsally and in the head fronts. They are shifted to the vertex in Diptera and Hymenoptera. The median ocellus is formed due to fusion of paired ocelli, or in some cases, the dorsal ocelli are completely degenerated. The basic difference between the ocelli and the compound eye is that the ocellus bears a dome-shaped common lens for all visual cells, while in the compound eye, each visual cell is provided with an independent lens. Lens is cuticular. Then retina is made up of only 4 retinular cells. Axons form ocellular nerve. Ocellular nerve terminates into the pars intercerebralis. Cord is absent. Pigment cells are absent, and the ocelli are incapable of forming image. The ocelli bears a single lens, and in most of the insects it is cuticular in origin, but in Epimeroptera it is formed from the underlying cells, cornegian cells, rather than cuticle. In Periplaneta, a simple transparent cuticle is present in the place of a thickened lens. Beneath the lens is present the epidermis, which is composed of transparent colorless small cells often called the cornegian cells. Beneath the layers of cornegian cells are arranged large number of comparatively larger cells, the nerve cells called the sense or reticular cells, forming the retina. Some of the reticular cells may extend from cornea to the base of the ocellus. The reticular cells are arranged in a group of 4 cells each, and the rhodomers of them form a rhabdom. They may contain pigment. The fibers of the retinal cells run backward and merge in the ocellular nerve, which terminate into the mid-dorsal region of the pars intercerebralis of the brain. They synapse with the neurons of the region. The inner part of the retina is traversed by a membrane of connective tissue or by durate crystals, cockroaches, forming a reflective tapetum. In some cases, peripheral corneal cells contain a dark pigment appearing as an iris. Moreover, a vitreous refractive fluid is present between the cornegian and the retinal cells. The ocelli are simple light-sensitive or stimulatory organs activating immediately the central nervous system, and this play a key role in maintaining the diurnal rhythm. They are well adapted for quick perception of light changing in its intensity. Then they are, however, mostly incapable of forming images because of wide angular separation of the rhabdom, and as a result of images are focused far behind the retina. Then the stem. The latter is follow metabolic larvae and some other genetic polarity naturally. Anopleura are more developed than the dorsal ocelli but less than the compound eye and are commonly called as the labyrinth eyes or stomata. Some workers consider them as the precursors and others as the degenerated forms of the compound eye. The stomata vary in number from a single pair to the 6 pairs among the holometabolic in nature forms. Adult insects often bear a single pair of stomata on the head structure. Each stomata is formed from a small group of photoreceptive structures. Maximum number exceeds 50 in the locus rosa. Each photoreceptive structure unit of stomata consists of an external endocuticular cornea, crystalline lens, which is flattening that of tetrahedronid, group of 7 retinal cells and some core membranes. The retinal cells differentiate based on distal retina, opposite cells forming a distal retina, and the proximal retina composed of 4 cells forming a distal retina. When all retinal cells are extremely superficially covered by the retinal membrane, and the microclear retinal supporting GM cells. The narrow fibrils of retinal cells enter the opticloser lateral part of the brain. Adult insects, each stomata is composed of cone bicycle structures using formula pluriangulated functionally, while 50 in some structura. Photoreceptor organs of muscoid maggots accounting to some markers formed due to degeneration of stomata. Stomata can form images on the rhabdons. The images are found on the proximal rhabdon when the object is closer within the distance of 2.08 cm and are formed on the distal rhabdon when the object is at long distance. They can differentiate the shapes and colors of the object. Caterpillars form mosaic images. Tubercle, epidermis, crystalline, nucleus, sense cells, envelope membrane, medium cells, distal, then the compound eyes. The compound eyes have a few visual organs like Pinera, Ectopoda, and majority of the pellicula. Compound eyes are replaced with stomata in the larger form of holometabula and convergence. This is an update of Mirapola actually to the direct representation of Lycan aspera, Symbolica, and Specific. They are generally reduced to the genus Pseudolinthitis and steroid forms of social parasites. The compound eyes are composed of a large number of light structural units called cones. The compound eyes typically multiply of stomata and division of cornea into large number of faces often equal to that of hemisphere. From the faces, the total number of hemisphere on a compound eye can be eliminated, which are known to vary greatly and from one interloper and Hornera scientifica. The faces are arranged in hexagonal form within few insects. When the number is less, they are circular in shape. Generally, all faces form a single spherical eye bulging out on the sides of its own side. The head is they may differ in two areas due to variation in the composition of the two regions are collected and technology. The faces size is mostly proportional to the square root of the height and the size of the face is situated at the center and it is from there situated on the origin. Stomatidium in stomatidium does not size and sexual multiplication of the second species dioptric or optical apparatus between the or the sensory or retina formation. The optical apparatus is composed of a reticular lens. The sensory part is composed of a lens specialized in vertical or the reticular cells. They form a random and the similar pattern of the stadium. The reticular cells are symmetrically represented of the cone of the primary pigment. The retina is separated at the bottom radiation membrane through which the axons are different from the retina pass into the optic lobe. Corneal cells, crystalline cone, retinal cells, pigment cells, laminar ganglion. Nucleus of structure cells, primary pigments, corneal lens, crystalline cone cells, cornegian cells, then retinal cells, secondary pigment cells, base nucleus, axial filament, rhabdom, nervous types of bacteria. On the basis of modification of cone, ommatidia are recognized in 4 types: eucone, pseudocone, acone, and exocone. The eucone ommatidia. They contain a true crystalline cone as a hard rectangle formed from an intracellular secretion of the cone cells of central cells. The eucone ommatidia. They contain a true crystalline cone as a hard rectangular body formed from an intracellular secretion of the cone cells of central cells. Lysinura, Orthoptera, Odonata, Echineroptera, Trichoptera, Hymenoptera, and Coroptera. Then the pseudocone ommatidia. They do not contain true crystalline cone, but all cone cells are filled with gelatinous semi-liquid substance. In the acone ommatidia, they do not contain either a crystalline or liquid cone, but the cone cells are illuminated and transparent. Lermoptera, Hemiptera, and another. Eucone, pseudocone, acone. Then the exocone ommatidia. They have an extracellular cuticular cone in the place of crystalline or liquid cone secreted by the cone cells. It is formed as an inroad from the inner surface of corneal cells in front of the unmodified cone cells. It is refractile in nature. Simply on the basis of rhabdom, the eyes can be classified into 2 types. Apposition eyes. These contain the rhabdom, which is very long and extending from the lens to the basement membrane, cockroach. Then superposition eyes. These bear a comparatively short rhabdom. It lies far behind from the lens to which it is connected by an axial filament. Structure of the ommatidium as described above. The ommatidium consists of the cornea, a cornegian layer, and the corna or Semper cells, and the primary and secondary iris cells, and the retinal cells. The cornea. It is outermost part of the ommatidium. It is transparent, colorless, and biconcave, convex, modified cuticular area, often termed as a facet or lens. It is cast off at each ecdysis and re-secreted by the epidermal cornegian cell. The cornegian cells, they are the epidermal or hypodermal cells lying behind the cornea. A group of 2 cornegian cells secrete a single lens. After formation of the lens, they withdraw peripherally and modify into the primary iris cell, the cone or Semper cell. Beneath the cornegial layer of cornea, there are 4 distinct cells. They secrete the crystalline eucone or liquid cone, pseudocone. The cone cells may simply occur as the transient cells without secreting any kind of cone, acone, or remain unmodified, exocone. In most cases, these cells are represented by the nuclei only and are called the Semper's nuclei. The reticular cells. The crystalline cone is followed by long retina, forming the basal part of the ommatidium. It is formed from the group of anaric and retinal cells and pigmented visual cells. The retinal cell posteriorly terminates above the basement membrane and gives out a post-retinal axonal fiber tending towards the optic lobe. The inner margin of the retinal cell lying around the ommatidial axis is highly differentiated from the rest of the body, and it is called a rhabdomere. These rhabdomeres are 7 retinal cells that extend the whole length of the retina and form a rhabdom. The rhabdom is nothing but an internal optic road having a fibrillary structure, and it becomes a central axis of retina. It is in distinct contact with the lower extremity of the cone in opposition eye, while lies far behind the superposition eye. The rhabdomere contains large number of microtubules or microvilli. Microtubules are about 400 to 1200 angstrom in diameter, hexagonal, and are extending towards the central axis of ommatidium and long axis of retinula cells. They are closely packed and all are parallel to each other. The diameter of rhabdom appears to be 0.5 dm in sarcophaga, while 1.2 dm in dorsum. Because the rhabdomeres are closely opposed against one another, forming a solid rhabdom and columella, Orthoptera, Lepidoptera, Hymenoptera, are fused in pairs, while in Diptera and Hemiptera, the rhabdomeres are also subject to When the rhabdomere contains large number of microtubules or microvilli, the microtubules are about 400 to 1200 angstrom in diameter, hexagonal, and are extending towards the central axis of ommatidium and long axis of the retinula cells. They are closely packed and all are parallel to each other. The diameter of rhabdom appears to be 0.5 dm in sarcophaga. So the rhabdomeres are closely opposed against one another, forming a solid rhabdom. Then the rhodopsin. The rhabdomere is the site of the chemical reaction. The common photoreceptor pigment is a chromoprotein called the rhodopsin. It is a complex of retinal and proteinopsin. The retinal is a carotenoid pigment, an aldehyde of vitamin A conjugated with the proteinopsin. The light adapted condition, the proportion of vitamin A to retinal is 1:4, and the dark adapted condition 4:1 due to hydrolysis, bleaching of retinal to vitamin A by hydrolysis, and as a result impulse is produced in retina fiber. When hydrolysis of retina does not occur, it accumulates due to continuous synthesis in the rhabdomeres. In Notonecta, not only pigment but the rhabdom also moves. The accessory pigment are pterins like prosopterin, neodrosopterin, sepia, etc., derived from riboflavin, and homochromes like vermilion and cinnabar of red, yellow, and violet pigments derived from the amino acid tryptophan. Chemical change in the pigment in rhabdomeres of ommatidia after light exposure occurs. The signal transduction is changing light energy into electrical energy. It causes depolarization of the retinal cell. Pigment or iris cells. There are two groups of iris cells, one around the crystalline cone cell and the other around the retinula, called the primary and secondary iris cells respectively. Each group of iris cells is composed of 6 cells arranged in a sort of circlet. The secondary iris cells separate one ommatidium from the neighboring one. The iris cell is provided with two types of pigment granules, black which absorb the light and pale or other colored granules that reflect the light. The pigment prevents the entry of the oblique rays, and by moving in the up-down direction, it maintains the illumination of the eye approximately constant with rapidly changing light intensities. The pigment of the iris cells in fact expands and contracts in accordance with the intensity of illumination without causing any change in shape or location of the cells, but the contraction and expansion results due to clumping or dispersing of the granules of the pigment. In butterflies and moths, the high intensity of light during daytime initiates expansion of pigment granules, while low intensity of light during night causes contraction of the pigment so that apposition and superimposition images are formed respectively. Tapetum. The long branches of trachea invest the basement membrane, forming a tapetum. It reflects back the light and as a result in the rays retraverse the rhabdom. It is well developed in the nocturnal flying insects, and in some insects, however, such as Apis, trachea are not investing the basement membrane. Optic nerve fibers. The optic lobe of each side of the brain is directly connected to the compound eye. The nerve fibers arising from the retinal cells decussate or run parallel and are of two types: short fibers running to the lamina and the long fibers to the medulla. The non-monopolar cells of peripheral region of lamina make dendrite synapse with the retinular fibers and their axons run toward the medulla within the optic lobe. Outram earlier classified the eyes into two types, slow and fast on the basis of monophasic and diphasic electroretinogram respectively, but comparative studies on insects and 8 orders reject such type of classification. Then physiology of the compound eyes. The optical system. Long back, scientists proposed the mosaic theory, stating that the individual ommatidia of a compound eye form an image in parts limited to their visual fields with different intensities, and juxtaposition of all these gives a total erect image of a complete picture of an object. Each ommatidium has a wide field of vision, forming 20 degrees Celsius in Locusta and Calliphora and about the nerve fibers arising from the retinula cells decussate or run parallel and are of two types, short fibers running to the lamina and the long fibers to the medulla. The monopolar cells of peripheral region of lamina make dendrite synapse with retinular fibers and their axons run towards the medulla within the optic lobe. Outram earlier classified the eyes into two types, slow and fast on the basis of monophasic and diphasic electroretinogram respectively, but comparative studies on insect or 8 orders reject every type of classification. Then comes the third one, that is the fourth one, physiology of the compound eye. The optical system. Muller proposed the mosaic system, stating that the individual ommatidia of a compound eye form an image in parts limited to their visual fields, which differ in intensities and juxtaposition of all these gives a total erect image of a complete picture of an object. Each ommatidium has a wide field of vision, about 20 degrees in Locusta and Calliphora and about 20 to 30 degrees in Apis. There is therefore a chance of wide overlapping in the visual fields of neighboring ommatidia, but the intensity of illumination falls off sharply as the angle of incidence increases, and thus most of the light entering the dioptric apparatus comes from a limited area of 1 to 2 degree ommatidial angle. Maximum intensity of light penetrates along the ommatidial axis. The dioptric apparatus of the insects possesses the lens cylinder system having special optical properties. It was first discovered by Exner in 1891 that the dioptric apparatus of the insects possesses the lens cylinder system having special optical properties. The dioptric apparatus resembles a system composed of a series of cylinders or cones superimposed on one another with their spaces keeping inward, so that the refractive index of the apparatus is maximum at the central axis, but it diminishes towards the periphery. The diplotric apparatus is moreover distinct in one more respect. In some insects, the focal length of the lens cylinder system is mostly equal to the length of the cylinder, while in some other insects it is nearly half of the cylinder length. The formation of the image therefore differs in both the above cases. In the former condition, it is formed at the posterior or retinal base of the cylinder, while in the later case, the image is formed at the midway along the length of the cylinder. These are the apposition and superposition methods of image formation. The mechanism of image formation. There are, as discussed above, two principal mechanisms of image formation, the apposition and the superposition. The apposition mechanism. It is a mechanism of image formation in the bright daylight in most of the diurnal insects, Odonata, Orthoptera, Hymenoptera, Diptera, butterflies and various Coleoptera. The pigment cells envelop the central point of the dioptric apparatus. The other peculiarity of the ommatidia of these insects is that of the focal length of the apparatus is equal to the total length of the cones. The formation of a reverse image therefore take place by virtue of the light entering from comparatively smaller part of the visual field, and that too at the retinula where it comes in immediate contact with the cone. The apposition of all such individual reverse images of different materia finally produces a single complete direct image of an object. Lens, cylinder, focal point. Rays, lens, pigment, wrapped. Mechanism of image formation, the apposition and superposition. The superposition mechanism. It is a mechanism of image formation in poor light, generally during night, mostly in the nocturnal insects such as Lampridae and moths. The secondary pigment cells are adapted to migrate themselves, for their pigments move in both proximal and distal directions. Particularly in poor light, the pigment contracts so that the light from the wide field area can enter obliquely without being absorbed. Each rhabdom therefore receives the light from large number of dioptric apparatus of neighboring ommatidia besides its own facet. The ommatidia are comparatively longer than that of an opposition eyes, and retinulae lie far behind from the cones connected by the non-refractile filament. The optical system of such ommatidia has therefore the focal length about half the length of the eye's lens cylinder. The image thus is formed from a group of rays refracted from neighboring cones called the superposition image. It is erect in position and it represents merely a part of an image. The compound image is formed after an amalgamation of all such images and retains a final form of complete superposition image. Diffraction mechanism. It is superimposition images at progressively greater depths in the retina of eye due to diffraction from the lens system besides the first apposition image. There are diffraction images which are formed due to diffraction of light received from the adjacent facets. The diffraction images are very complex and distorted in comparison to either apposition or superimposition images. Their functional significance remains unknown. Pigment movements and eye adaptation. The pigment movement occurs in the opposition eyes of butterflies and hemiptera, in the superimposition eyes of most of the nocturnal insects, so that in accordance with the availability of light, the same eye can act as the apposition or superimposition. Due to such adaptation, some of the nocturnal insects are active in the day and perform their activities perfectly. The apposition eye may act as a superposition eye in the dim light if the pigment of the iris cells is withdrawn. Similarly, the superimposition eye may act as the apposition eye in the bright light if the iris pigment expands downward surrounding each retina as a black curtain. The adaptation may take a period of a few seconds to some 10 to 15 minutes. In man, the anterior part of the eye is adapted to apposition while the lateral parts are to the superposition. In perception of colors, wavelength discrimination in insects follows trichromatic theory of color vision. The trichromatic receptor system is well maintained in the honeybee with independent maximum sensitivity for each receptor without Purkinje shift. The highest sensitivity appears for the green for superposition, dark adapted eyes, and for the blue for apposition. Apis can discriminate 6 colors: yellow, blue, green, blue, violet, ultraviolet, these purples. However, discrimination of ultraviolet, blue, and yellow occurs much faster than other colors. The primary colors discriminated by Apis are therefore ultraviolet, blue, and yellow, while the complementary colors are violet, blue, green, and these purple. The insects cannot discriminate red color and have red-green color blindness like that in a man. The red color of flowers is discriminated by the insects due to mostly the ultraviolet reception. In man, blue, violet, green, and red are the primary colors, while in blue, green, yellow, and purple are the complementary ones, and it therefore seems to be different from the man in vision discrimination. Color vision is known to occur in various insects like Hymenoptera, Diptera, Coleoptera, Lepidoptera, Neuroptera, Hemiptera, and Orthoptera. Ultraviolet, B purple, B violet, yellow, blue, blue-green. In human, blue-violet, purple, blue-green, yellow. Perception of form. The objects of various forms like solid triangles, squares, circles, and ellipses are indistinguishable from one another by the insects, but can easily be distinguished from these many figures after breaking into black or white areas like crosses or checks, hollows, spheres, rows or stripes, or flower-like figures. The insects are able to differentiate objects with an angular separation of 1 degree or 2 degree, and thus larger objects are readily visible to the insects. They can quickly distinguish the objects having contrast background. The compound eye is more adapted for the perception of movement rather than the form. Visual acuity. It is a measure of a pattern resolving power of the eye, which depends upon the dioptric apparatus and retina. The visual acuity of insect eye is 1 by 60 to 1 by 80 of normal human eye. The visual acuity of the human eye is 2.0 to 2.5. The insect eye is capable of resolving two objects with an angular separation of about 1 degree Celsius, while those closer together than 1 degree are not distinguishable from each other, and thus it represents minimum visual angle, which is the measure of visual acuity. With the diffraction image formation, the insect may, however, differentiate objects with an angular separation of only 0.30 degree, Locusta and Calliphora. The visual acuity is affected by the intensities of light and the density of functional elements in the retina. The acuity of compound eyes is discriminated by interromaterial angles, optical duality, and random dimensions. It is also affected by light levels and speed of movement. In insects, interromaterial angles vary from tens of degrees in Apterygota, so as little as 0.24 degree in dragonflies, providing the maximum resolution, and the smaller the interromaterial angle or greater the distance at which objects, prey, predators, and pelagian are resolved. Insects with different lifestyles have constructing patterns of interromaterial angle distribution related to forward flight, capture on the wing, and predation of horizontal surfaces. In an electroretinogram and fast and slow eyes, the electric recording of the eye is called the electroretinogram. It represents a sum of potentials arising on the optic lobe and eye in which the cornea is negative in relation to the optic lobe. The generator potential, it seems to be exclusively derived from the retinular cells. The generator potential shows positive one effect with the loss of illumination, showing a short-lived change and transient change in potential respectively. On the basis of ERG, Ottrup has classified the insect eyes into two types, the first fast eyes and the slow eyes. When our ERG records diphasic wave consisting on an on effect and an off effect repeatedly, the eyes are called the fast eyes, which are reported in the fast flying insects such as Diptera and Hymenoptera. The fast eyes are found in the diurnal insects and are less sensitive to the light. They have the flicker fusion frequency of about 300 times per second. Due to sharp positive on effect, sustained depolarization of retinal cells is prevented, and as a result, they respond to the faster moving objects. But when our ERG records a monophasic wave of potential showing a sustained, slowly diminishing response to illumination, and in addition, when the amplitude of wave varies with the light intensity, the eyes are called the slow eyes. These eyes are found in the nocturnal and slow flying or walking insects such as Orthoptera, Plasmida, Holoptera, Leptoptera, etc. They have poor discrimination of flicker, as their flicker fusion frequency measures about 40 to 50 times per second. They are, however, more sensitive to light in comparison to insects with a fast eyes. Due to the presence or absence of screening pigments, red-eyed and white-eyed Drosophila melanogaster have electrorethiograms with different sensitivity spectra. The same differences were found in a comparison of ERGs of red-eyed and white-eyed retinal degeneration mutants. No effect of the pigments can, however, be found in the spectral sensitivity of escape phototaxis behavior. The observations imply that only receptor cells and on-axis ommatidia contribute to this behavior even in the white-eyed fly. In the holometabolic and hemimetabolic insects, the growth in size of the eye which takes place by the addition of new ommatidia is accompanied by specific decrease in the amplitude of the electroretinogram. The ganglionic response of the holometabolic insect appears only after the adult is fully mature, but it appears as early as the second instar in the hemimetabolic insect.